Hello, can you hear me okay? Tommy, thank you. Okay, good afternoon, everyone. What a great turnout. Thank you for being here. Welcome to our friends who are watching online via Wisconsin Eye today. Welcome to Promise and Perils of Social Genomics, presented by the Follette School of Public Affairs in partnership with Wisconsin Health News. For those of you who may not know me, I am Lisa Ellinger. I am currently Follette's Director of Strategic Initiatives. I'm also a proud alum of the school. And I'm here today because I actually spent most of my pre-Follette career working in health policy, and I was thrilled to see names from my past lives at the Wisconsin Hospital Association and Department of Employee Trust Funds, and my current role serving at the Access Community Health Center's Board of Directors. Great to see some friendly faces and names on the list. But the reason I wanted to bring that up is, having spent a good chunk of my career in health policy, I'm absolutely thrilled about this topic and these researchers and this event, and I'm honored to be here to kick things off today. So over the next hour, what we're aiming to do here is to provide a little bit of level-setting Social Genomics 101. It's a groundbreaking field focused on interdisciplinary research that integrates the social sciences with genetic research. It aims to show how our genes are linked to behavior and socioeconomic outcomes. It researches how our genes and environments interact and how social conditions can influence humans all the way down to our cellular level. And all of that is made possible by amazing recent scientific breakthroughs in genetics and the data and analytic tools that are constantly evolving and being developed and refined to study this new frontier. So I'm probably stating the obvious here. The implications of this research could be so profound, and it is allowing us to be able to address questions where previously we could only theorize. So now we can dig deep into our genes to see how our environments, impact us, and that holds great significance for so many other areas, ranging from public health policy to precision medicine to poverty research, longevity research, and I could go on and on and on. So to help us wade through this fascinating and important topic, we have three leading scholars with us here today. You all have their bios in front of you, so I will be brief. But I'm very proud to introduce Lauren Schmitz and Jason Fletcher, who are colleagues of mine at the Follette School of Public Affairs, the amazing Anya Prince from the University of Iowa's College of Law, and last but not least, our moderator this afternoon, Tim Stumm, who is one of Wisconsin's leading health journalists and founding editor of the Wisconsin Health News. And Tim, on behalf of Follette, we so value and appreciate your partnership. So thank you for helping us make today's event happen. So before I turn things over to our next speaker, I just wanted to remind or make this group aware that this panel kicks off a two-day conference called the Advances in Social Genomics. And over the next couple of days, we have more than 24 leading researchers from around the world sharing their cutting-edge research with each other right here on the UW-Madison campus. And this conference is made possible by the Follette School's initiative in social genomics, which is also supported from the National Institute of Aging and the Center for Demography of Health and Aging. And the initiative in social genomics, if you are not familiar, is the largest group of researchers pursuing topics in social genomics in the world. And it's all happening right here at UW-Madison and the Follette School, and I just think that's super cool. So thank you for our leaders on that. Final reminder before I turn things over, the panel is streaming live on Wisconsin Eye. Again, thank you, Wisconsin Eye, for your partnership. And if anyone in the audience knows of a colleague who can't be here today, feel free to send them a message and let them know that they can tune in. But we will be recording this panel and the full conference and make all those recordings available online after our in our post-conference communications. So, before Tim leads us through a thought-provoking and insightful Q&A session, I'm going to turn things over to Lauren Schmitz from the Follette School, who will briefly set the stage for us with a little Social Genomics 101 intro. And Lauren is not only one of the world's top researchers in this field, she's also a skilled science communicator. So please join me in welcoming Lauren Schmitz.
Thank you for that fantastic introduction, Lisa. And thank you all for being here today. It's really exciting to see so many faces in the room. In February 2001, a while ago, a working draft of the human genome became available. And by 2003, the first map of the human genome was published. Today, over 2 decades after the human genome was first sequenced, we have many more questions than answers about what makes us tick. We stand in an era where rapid technological advances in computing and genome sequencing have produced an ever-expanding fire hose of genomic data that we, as a collective scientific community, are still trying to make sense of. In part, sequencing the human genome wasn't the silver bullet humanity hoped for, because we realized that we can't study the human genome in isolation. If we want to gain a better understanding of how genetic diversity shapes who we are, we need to understand and get outside the lab to study genetic diversity and our genes in the wild. After all, we are social creatures living in a complex societal ecosystem, and these ecosystems, from the air we breathe to the job we work, also powerfully shape our life outcomes. So in short, we realized that we need to observe not just nature or nurture, but also the interplay between them. And I think that's really what this discipline, this newer frontier, seeks to do. UW-Madison has become a leading hub for interdisciplinary researchers who are exploring this complex interplay between our social and cellular worlds. The initiative in social genomics at UW-Madison brings together a group of economists, statisticians, sociologists, psychologists, and epidemiologists, all with additional graduate research training in genomics to engage in cutting-edge big data research. Today's panel will showcase some of the questions we're investigating as researchers and why these questions and their answers are particularly important from a healthcare and public policy standpoint. So without further ado, I'm going to hand over the mic to our moderator, Mr. Tim Stumm. Thanks so much, Tim.
Well, thank you, Lauren. Some great words to get us started on. And thanks, just want to say thanks to Follette for asking Wisconsin Health News to be part of this really great event. I myself am excited and look forward to learning a lot about this really important topic. I do want to note real quick that we are going to leave some time at the end for audience questions. And I know the experts and myself are really looking forward to that part of today's event, so please start thinking of those questions now. And so let's, I'm just going to dive into the questions and get going here, but Jason, let's start with you. You may be too modest to say it, but you really are one of the pioneers in this emerging social science. Can you tell us a little bit about the promise of social genomics that appealed to you when you began doing this work?
Yeah, so Lauren, thank you, Tim. So Lauren really set this up nicely. Even when she was talking about genes and environment, I saw some nods in the crowd. So I think that's actually a key point that social genomics really insists on. It's not nature or nurture, but it's nature and nurture and their interplay. But that insistence really complicates everything. You could kind of look back and say how useful it could be if we could silo ourselves off and only be trained in genetics and find the genetic effects of important outcomes, and they're really big and really clear, and we could even create treatments for them. And then separately, on the social science side, we could only focus on the environment and try to move genetics off the table as much as possible and think about the best policies for whatever we're interested in. But it turns out, and many of you, as you were nodding, both believe this and have experienced this in your life, that it's both. And that complication, though, both, I think, allows us to think we're on the right track to say it's both, and we need to really investigate both. But when you then try to do that, when you try to and do that well, you're really confronted with a lot of roadblocks and hurdles. And those include going back to how we train people in graduate school in particular to specialize, to focus on your one thing and try to change the world in your one domain, and really otherwise remove everything else off the table. But in doing so, if you can kind of get it wrong, because you're by focusing on your one domain, you're not including all the relevant factors. So these complications and these roadblocks come from both data, which until recently has been very siloed itself. There was social science data collected by social scientists for social science enterprise, and there was genetic data in a different path. And it's only been in the last 2 decades or so that there was even there's even been a beginning of both those data elements being in the same data structures. And as Lauren just said, we kind of went from zero to fire hose very quickly, though, where now we're way off from having the concern of, oh, there's no data that's relevant for me. It's just what can I do with all the data that's available. But in thinking about what can I do, the other obstacle has been essentially personnel. Who is it that are trained to be able to take to wrangle this fire hose of data, because they somehow know something about both environments in social science and genetics? And I think that's where we are making major investments in having, as Lauren just said, people who are willing to have their first training first, and Lauren and I are both trained as a social scientist first and then added on later, but many of our colleagues at UW have done the reverse, where their basic training is in genetics, and they're willing to be kind of cross-trained with us in these workshops and conferences. And so I think that's the excitement, that we feel that this integration is putting us on the right track in terms of understanding the world in the way we want to understand, in a way that we could not if we retain those 2 lanes of everybody being trained on one or the other. But it's also, as we'll, I think, hear and want to hear from the audience about questions in this domain, it also puts up a lot of complications, because it is so complicated. The solutions so far have not been obvious. They've required a lot of work, and we're not there. We don't have the solutions yet, but I think that's the enterprise here, is that we need collaborations to build this bridge where both sides are building at the same time and coming together. And I think that's that's the promise of work here and in many other places. So I'll stop there.
Great, thank you, Jason. So Lauren, social genomics encompasses a lot of different research topics. Can you just talk a little bit about some of the questions that you're trying to address in your research?
Yeah, thanks, Tim. So these days, my research is primarily focused on aging and longevity. So, and in particular, how our social circumstances and social disadvantage can accelerate or decelerate our biological age. So we've known for a long time, gerontologists have told us for many decades, that our chronological age can differ from our biological age. And we see this kind of anecdotally, again, in the wild out in society. We might see people who are our exact same chronological age, but who look decades older sometimes, or 10 years, a lot older than we do. And we see some people who look much younger. So we know some people are aging faster biologically than they are chronologically. And now, with epigenetic data, we can actually calculate people's biological age quite accurately. So this is a whole nother layer of data on top of our DNA. So we know our kind of simple DNA code. And then on top of that, we have these chemical modifications and tags called epigenetics. And in particularly, these clocks use an epigenetic phenomenon known as DNA methylation. So this is whether these certain chemical tags are attaching to our genome, and then they're either upregulating our genes and telling them to express, or they're downregulating those genes. And so over the past decade, scientists have noticed that these chemical modifications and the changes in these tags over time are actually highly associated with our biological age. So by just taking a simple blood sample, we can calculate somebody's biological age. And then we can see what circumstances in their life might be accelerating that aging process or again, decelerating that. And from a policy standpoint, we care about this because these measures are much more sensitive. They really allow us to see how public policies or different interventions might be accelerating people's age or decelerating. So if you see age acceleration, that typically is associated with faster, earlier mortality. So this kind of, again, this scientific explosion of data is really allowing us to see the impacts of public policy at a cellular level. And from my own research, an example of this paper that I had that came out back in 2022, co-author and I looked at how the Great Depression shaped how fast people aged biologically. And in particular, what we found was that individuals who were in utero or in the womb during the Great Depression were aging faster decades later. And so here we see this really important connection between early life conditions and late life aging. So again, this is something that's only now possible because we can bring together social data, historical data, with data, again, on our complex molecular worlds.
Great, thank you. Jason, could you also just kind of follow up on that? Could you talk just a little bit about some of the other big initiatives that have come out of UW so far in this field?
So Lauren is certainly leading on the social epigenomic side, which is the epigenetics, the environment, switching, tuning up our genes, tuning them down, but with a focus that's somewhat specific to social scientists in their interest in both historical. Great Depression, of course, is a major economic event, right, of very big and policy importance. And so Lauren is very much leading up that wing. But we also have several researchers examining the molecular genetic variants that are now no longer the epigenome, but the A's and C's and T's and G's in our genetic code, and asking how those are represented in the outcomes, in our health outcomes and our socioeconomic outcomes throughout our life. And both from a predictive exercise where we could maybe take everybody on this side of the room and you would all spit in a cup and we would all genotype you and get all your A's and C's and T's and G's and ask whether, and then also measure your, whether you're blue-eyed or brown-eyed, and also your height. And then for every one of those A's and C's and T's and G's, every location on your genome, we'd ask if we see an A there, is it more likely that they're blue-eyed and tall, or the opposite, or they short, and then keep on going throughout the millions and millions of variants across the genome. And each of those little variants for eye color, you'll see this interesting architecture where only a couple of those A's and C's and T's and G's matter, and they matter a lot. And on the other hand, for height, which is the second thing we measured, there's no one letter that really matters, but you have to add them all up over the genome to get a predictive measure for height. So we have simple outcomes like eye color, and very complicated outcomes like height and many health outcomes. And in each of those cases, we would take this data set and create these predictive measures and ask, over here, who are not in the data set that I'm describing, how predictive is it for you? And one of the major topics of interest here are to make those predictions more accurate. And the major topic on the table, the major hurdle on the table, is the lack of accuracy, depending on how similar you on this side of the room look and are and where your families came from as compared to the families on this side of the room. And to the extent that you differ, those predictions are much worse. So we have a whole set of our colleagues really trying to attack that particular major problem. We'll come. I might be segueing into part of what Anya's talking about, but that prediction, that little cartoon I was describing, is what our hospitals want, what our clinicians want to be doing in the clinic, is to take genetic data and ask, are you high risk or not for heart attacks or for some other health measure? But that little exercise I was just describing also shows some downsides where if you're in the clinic and the data set that was used to make the prediction of these people over here don't look anything like you and don't have your background, then those predictions can be quite bad. But the clinician doesn't know that. So that's a first order problem that people here and across the world really are tackling. But I think I'm setting up Anya pretty nicely for
Excellent. Well, let's go to Anya. Anya, you have a pretty different focus on this. You are a lawyer specializing in genetic discrimination and the health privacy implications of big data. I guess you could just talk a little bit about what does that mean? What exactly does that mean in the context of social genomics?
Yeah, great. So thank you so much for having me. It's a pleasure to be here. So I think I'll start out with just talking about three things that I think about when thinking about social genomics. And one, Jason really set up well by pointing to you all, right? The data, the fire hose of data that we are talking about are people. So what the scientists are doing are taking hundreds of thousands and millions of people's genomes to calculate these scores. And we can do this for health outcomes like heart disease and cancer and all the like. But also with social genomics, you're starting to predict, as Jason measured, socioeconomic things. So things like your genetic predisposition to income. Predisposition might be the wrong word here, but your genetics association to your income or your educational attainment or your reproductive behaviors. Now, some of these are going to be on the medical continuum and some of them are more social. But what I think about is how do the biobank participants who signed up and gave their data think about this? Because a lot of people who put their data in biobanks think, oh, I'm signing up for health research. And they might be very surprised to take probably one of the most provocative examples to find out that there is a score created for same-sex sexual attraction. Right? And so millions of people's data has been used to create that score, and people might feel very conflicted about that if they had known that that's what their data would be used for. Now, I'm not insinuating that those scientists weren't using the data correctly. They got permission, there's consent, there's all of that. But you still wonder what people would have thought if they understood what's going on. A second thing that is one of my major focuses is we talk a lot about how this can be helpful for research. It's really helpful to understand gene by environment research when we can understand both the genes and the environment together. But what happens when that data and the findings are taken outside of the research realm and into society? So Lauren's research, super interesting about biological aging. You know who finds biological aging really, really exciting? Life insurers. They are on the ball and they are ready to take a simple blood test and test what your biological age is. Because if they could differentiate between a 45-year-old who chronologically is 45 but biologically is 65 versus a 45-year-old biologically who's actually 30 chronologically, done. They are predicting and they have a better insurance pool than their counterparts, right? And then the third thing that Jason was really mentioning that I think is important to think about in terms of the ethics and policy implications of this research are the equity impacts. And so one of the things about these hundreds of thousands and millions of people's data that's going into this research is the vast, vast, vast majority of it comes from predominantly European ancestry, wealthy, well-educated individuals. And so when we talk about it might not predict for you as well as it predicts for you all, it can be even worse than that in that already the people who, the communities that we don't have participating fully in our healthcare systems are those that might have the worst data, just the data that we don't know how to interpret. And so what does that do to our already bad health disparities if we're then not having the benefits that can accrue to all populations? So those are just some of the things that I think about with this research.
So thank you, Anya. That's really interesting. Can you talk a little bit, just to follow up on that, what are some of the safeguards that can be put in place, around this to make sure that it's not being misused?
Yeah. So, I think one of the challenges with this is balancing the good uses versus the bad uses. How do we allow this research to go on and to get the really important findings that we can get while stopping what people would think would be discriminatory outcomes? And I think one of the challenges of that is there might be very different opinions as to what people think is a bad use, right? So a couple of examples would be the polygenic score or a score, right? So you can take all of these samples and create a score or an indices, and then that's the score that's ported. I'm not sure if we've used that term yet. But you can take these findings, let's say for educational attainment. So there are some that say that could be really great for us to understand in the research context. And there are others that say that could be really cool to take into the educational setting and have like precision education. So the people who have lower educational attainment scores as shown by their genetics maybe could get more help. That's the utopian version. As a bioethicist, I always have a dystopian hat on because that's my lot in life. And so we think, oh my gosh, this is going to be horrible. You bring in genetics in the classroom and you have problems. But how do you know what's good or bad, right? So we could set up laws that say you can't use it in an education environment, but some people would say that's bad because if we could use it for good in the educational environment, how do we do that? But just like some couple of examples that are on the books. On the federal level, we have a law called the Genetic Information Non-Discrimination Act, or GINA. This was passed in 2008 in response to concerns of genetic discrimination after the Human Genome Research Project that Lauren was mentioning. And it prevents health insurers and employers from using genetic information. And so those two entities, and there's some caveats, but those two entities generally cannot collect genetic information from anybody, and then they can't use it to deny insurance, have higher premium rates, fire somebody, fail to hire. But there are many other organizations that don't have any protections. So for example, for the most part, life, long-term care, and disability insurers, that is regulated at the state level, and states have wide differences in how they regulate. So here in Wisconsin, there is a law that says that those insurers can use genetic information as long as they can show that it's statistically meaningful. Well, guess what? They probably can show that a biological clock research is statistically meaningful to differentiate. And so we could pass stronger laws like GINA at the state level that says insurers or other actors shouldn't use this information, with the caveat that not everybody is going to agree that that's a good thing.
Jason, could you just talk about how you envision that this research is used by public health professionals to improve people's lives?
So, kind of going back to what I started out with, I think it's true, and we probably all agree, and this is going back to the nodding in the crowd, of when we think about health and we think about clinical encounters, that a model where we deny either that genetics matters or that these important social environmental factors matter is not the model that we're interested in. That we need to continue, I think, correctly believing that the model is both genes and environment in their interplay. And so I think we should be enthusiastic that those beliefs and those experiences that shape those beliefs are now coming closer to being where the data can meet those beliefs, and we can, if we want to. This is very much if we want to. We can act on those with major caveats. But so directionally, I think we would say this is the right direction. More data, more understanding, more collaboration. But clearly, 100% being humble about what we know right now, and not think that we're ready now to scale up in every clinic using only the knowledge that we have now. So that's the goal, is to bring. I was going to say that that's most people's goal, is to bring this into the clinic in some way to target treatments in a better way, to sort people in some way where they might be a better candidate for one treatment versus the other, and both genetics and social factors could be part of that shaping. But I think the largest challenge are two. So I'll just briefly just say what Anya just said is one of the largest challenges. I don't need to repeat it. Just both the relative ignorance of the public of what's happening and how they might think about what's happening, and also our mostly continued siloing of expertise into genetics on one lane and social environment on another lane, and not making it, not insisting that they both work together in the clinic. So I think there's both. This is the promise and the peril guaranteed in the title, in my view.
So what about how that's how this could be used in the clinic and in public health and in health? What about in the policy arena? How can this work, this research be used for policy?
So Anya said exactly the right phrase of taking precision medicine and precision health and asking what's our appetite for precision policy, which is to say we're only excluding policies we're only excluding the areas of our lives in the U.S. anyway of health insurers and employment, but education on the table, life insurance, most of the rest of our lives are on the table, and there's important policies, which we call environment, that shape how our outcomes in education all the way through, our health behaviors and so on. So I think there's a question that the public really would need to be involved in a larger conversation about in what areas are we quite comfortable with bringing in these predictive measures to potentially shape children's placement decisions in kindergarten all the way up to what college you go to. None of that is illegal. All the way to our life insurers, car insurance, and so on, about what elements that the public would like to be discriminated around. And I think right now the decisions aren't made mostly because they're not even on the table, they're not being discussed. But I also would concur that it's not clear that that we would speak in one voice with these major elements. But right now it's kind of a wild west of we don't have the restrictions either side. We're not saying yes, we should do them, or you cannot. It's just let's see what happens now.
Anya, you kind of touched on this already, but maybe expound on it a little bit. I mean, what do researchers need to do to convey to policymakers, about these moral, ethical, and legal concerns? I mean, what advice do you have to make that argument, that conversation with policymakers?
Yeah, I mean, I think it's one of the most important things is to try to communicate the nuance of this all, and that is incredibly hard. Both because policymakers are busy and moving quickly, and also communicating with the public and all of that. But just to give you an example, one of the things we haven't said yet is how predictive this research is on some of these social traits. Now, for some outcomes, like educational attainment, it might predict upwards of 15, I think we're around 16% now for educational attainment. So if we said all of you have varying educational attainment, we probably have a biased sample here, but the variance in this educational attainment in this room, your genetics would help to explain 16% of that variance. But some of the other social predictors that we're having, that Jason mentioned, adding up all those little variants across score, are predicting less than 1%, 2%, 3%. And that can be helpful for research, because if you can control for 1% of the variance, that's great, right? You understand the outcomes better. But for policymakers or the public who might jump to genetic determinism to say, oh, well, I have this score for risk tolerance, right? I'm riskier, but actually it doesn't predict that. It doesn't help to explain why I'm terrified of hiking and my husband loves it, right? Near cliffs. I don't mind hiking, it's just the cliffs. Okay. So I think that we need to communicate that nuance to policymakers. And the other thing that is really challenging for policymakers is how do you create policy that is deliberate and thoughtful, but also adaptable. So let me just give you one concrete example. I mentioned the state law here in Wisconsin that regulates how life, long-term care, and disability insurers use genetic information. Well, they define genetic information in the law tied to a disease or condition. Well, if I have a genetic test for income, does that law not apply to that information now? Because income is neither a condition nor a disease. And so you might have actually, because the policymakers were writing a definition thinking of a genetic test for cancer or a genetic test for heart disease, they might have actually written out of the law social genomics. Now, you could make arguments, and as I think good lawyers would make arguments, that pushing it back towards the medicalization, but some things you don't want to push towards medicalization, right? Same-sex sexual behavior. That's not one that we want to say, oh no, that's really medical. That's a condition, that's a disease, right? You wouldn't want to say that. And so you might have you have to communicate to policymakers that we have to think really expansive and really carefully about how we draft policy in order to be adaptable for the advances that are going to come. and things that we haven't thought of.
Lauren, I want to take a step back and talk about building out this data and the importance of this. Can you talk a little bit about some of the work that you're doing in Malawi and this research, the social genomic work you are doing there, and how, why it is so important to build out this data?
Yeah, thank you. Yeah, and Anya did a great job queuing this up earlier when she was talking about the existing disparities in terms of the availability of genomic data. So we do have this overabundance of genomes from European ancestry populations and typically from high-income country contexts. So, for example, there are whole regions of Africa, for example, which is where all of our genetic diversity comes from, where we know very little, actually, about the genetic diversity. These people have not been sequenced. There's certain regions that haven't been sequenced. And it's really important to gain a better understanding of human diversity writ large, so we can start to develop therapies and so forth that work, across the spectrum. So I'm currently co-leading a large NIH grant that's collecting genomic and epigenomic data on individuals in the Malawi longitudinal study of families and health. So this is a long-running cohort study in sub-Saharan Africa in Malawi, which is a very low-income country. It's the 4th poorest country in the world. I think this is really important for the international scientific community because we need more representation in genomic data across populations, and also because this is a particularly low-income context. So this will kind of increase the amount of data that we have in these populations, and our long-term goal with this from a policy perspective is to yield more generalizable evidence for these folks. So for these people, for these millions of adults who are living in similar contexts with mostly subsistence agricultural economies and inadequate health systems. So, we might understand precision policy for our tiny little neck of the world, but do we understand how this might work in a very different context where people might not have access to the same resources? So we're hoping that these data that we're collecting will help to generalize information more for that particular region of the human population.
Great. So I'm going to stop there and open it up to the audience for audience questions. I think we're just going to say your question. I think it's a small enough room, we'll all hear you, and our experts will do their best to answer. Actually, we have a mic. Yep, that makes
Hi, thank you. This has been very interesting. So I heard a lot from you guys about why, a lot of negative reasons why we shouldn't use this information for social policy. Do you see, like one of that I can think of, we talked about like how these genes are associated with outcomes and how this can be confounded by socioeconomic status and some of these, so by say using these genetic indices or these epigenetic clocks for insurance purposes, for example, you might be double penalizing low SES, low income individuals, right? So I was wondering from the panel, do you see an example where clearly you do wanna, that might be a positive thing into using this data and with the caveats of like what we know about these associations, these relationships for policy? Is there an area where you think we're ready to move on?
So I think I can start and then happy to hear others. I think this gets to where I was talking about where people are going to disagree. So in the insurance context, insurers are in the business of discrimination. And I don't mean that in a negative valence, but when you apply for insurance, they are looking at your factors and then differentiating or discriminating against this. So I think if I asked a poll in this room, how many of you would think it's fair to charge smokers and non-smokers the same amount for life insurance? My guess is that we would have many people who would say, I don't smoke. I think it's fair that I pay less than the person who smoked, right? And we might have some people who think about, social determinants of health and all of that. But we could have the exact same conversation with epigenetic clocks, right? If I have a lower biological age because I've lived a healthy life and made decisions, then there are people who would say that is fair for me to pay less than somebody else, right? So that's one example that people will argue that that's fair. Another example where this is being used already, or to some extent, is in the reproductive context. And so there has been genetic testing offered by some companies in the IVF context to say that if you have a choice of embryos, and I'll just put a caveat that not everybody has the choice of embryos, so I just find that a frustrating part of that conversation. But if you have a choice of embryos, then why not know your score for educational attainment and be able to choose the better embryo? So that is one where it's actually being rolled out. And then the big one that is often talked about that we've hinted at is the educational environment. So if a score for educational attainment could differentiate somebody who has a high score for educational attainment but isn't achieving that, then you could figure out what barriers are in place that is not letting them meet their potential. Or vice versa, you could say, why is there this low genetics? Well, maybe it's because the association, one of those variants is associated with dyslexia. And then you could identify some condition earlier, give them the resources, and then they could thrive better. So there's just a handful of glosses on that.
I'm good.
Got it.
That's
Got it.
right.
All right.
Where I'd like more clarification on where that data is coming from. Are they targeted surveys and studies, or are you picking data from, say, hospitals, or is it a combination of all of the above? And how you relate information that was taken at a hospital or a fertility clinic, and how you relate that with not necessarily the environmental factors keynoted in that particular study. How do you cross-reference that?
Yeah, I could take a quick, a great question. The bread and butter of most of what I do right now, and this is because this is what's kind of most readily available and that best fits into kind of what I want to do empirically as a researcher, is I use a lot of representative population surveys that have also collected genomic and other biological data on their participants. So for example, the Health and Retirement Study, it's the largest aging study in the world, began back in 1998, actually because Congress said we need to study aging. People in this country are aging. So they've been following people longitudinally since then. In 2005, when it became cheap enough to genotype people en masse like that, they began to collect genetic data, and now they're collecting also epigenetic data on these folks, gene expression data. So the kind of amount of biological data now that we can collect from representative samples from just one vial of blood is just getting better and better and cheaper and cheaper. But for now, these kind of population representative studies are mostly what I use in my data, in my research.
But the larger question is so that's a great example of a snippet of where the data are coming from. But your point about everywhere is the answer in some sense. Like you could start in Iceland and say, what kind of data they have in Iceland? And it's like everybody in the country is in one data set. It's only 350,000 people or so, but they're all in one single data set. The UK, there's a large and very important enterprise of collecting many hundreds of thousands of people's information, where it's both survey linked to electronic health records with follow-up information and so on. So it really varies, but in the end, a lot of it gets kind of pushed all together in a single big data set to go through and look for where these A's and C's and G's and T's I was describing earlier for a large population. I mean, closer to home, there's All of Us data is one of the major United States enterprises that has a site here, an important site here at UW, and their goals are really, they're enormous, which is, collecting over 500,000 individuals who volunteer to be in the study, complete a lot of surveys, but they contribute biological specimens that could be saliva and blood and others to get to be part of this fire hose of genetic and biological information, all linked to electronic health records and the survey information. There's some hiccups, or there's some bumps in the road here where the last federal budget essentially cut 80% of the funding for all of us. So it's unclear what's going to happen in the future. But the promise is really important because, as you've heard several times, that the representation of essentially non-white populations in the world is really small, and they're way underrepresented. But the U.S. happens to have a very rich melting pot of and so one of the keys was to enroll tens, if not hundreds of thousands of individuals who weren't European ancestry to build that data set. And so it's a super important enterprise. It's just hit a bump. It's not unclear how big this bump is right now, whether it's going to be a wall or not.
Just to give a very quick example as well, some researchers use 23andMe. So if any of you have done 23andMe and then filled out the questionnaires, like you're bored at night and you're answering, like, what time do I wake up at night and do I sleep well and all of that, there's a study for you. You get 23andMe and you get that data. So just an example of that some of your data might be in.
The difference being you've paid to be a research subject in that sense.
Yes.
Good question in the back.
Hi, I'm Tim Sweeney. I'm a faculty member here, and decidedly this will show you the biases. I'm from the social sciences side. The reason that we collect all that information in all of those longitudinal surveys and then add genes is that you start out with all the understanding you have from everything else that's in there, then you can say what difference did genes make. And we're not stopping with genes, of course. We have microbiology coming on next, what you've been exposed to, what you eat, and so forth. But let's just stick with the narrow version that you have now. What would you recommend to a geneticist who wanted to start where they were and then move to the social sciences side? I mean, I do believe that, there's a lot of cross-learning here, and I understand the path that I'm sure Jason and, and I'm part of that club, and all that. But what about the other guys? What would you suggest?
I mean, I think, and this is where I think we have such a wonderful group here, is that I think, doing more interdisciplinary research. So being open to collaborating more with scientists outside of your field. And I think that's where I've also really learned from other geneticists, other social scientists and so forth, is through collaboration. So I think that's one good part. You can say, hey, I can bring this to the table. I want to learn more about how this aspect of the environment affects things. Can you help me bring that to the table? And I think that's what we're going to need to do in this era of big data, is just have really large cross-disciplinary teams to answer some of these questions, because they do require bringing together so much different expertise. Within genomics, too, there's researchers who specialize more in wet lab-based work, and there's researchers now who do everything on the computer, who are entirely computational in what they do, because, again, we have all these big data now on people's genomes. So we're not confined anymore just to the lab and just to mice. Now, that work is also extremely important, but we can also now kind of collaborate, I would say, more easily with computational geneticists, statistical geneticists, bioinformaticians, et cetera, because we're kind of all speaking the same language in a way, which is big data. So it's kind of just about, okay, let's talk about how we can bring these data together and ask some interesting questions. So I think that's one way.
Hello, thank you for taking the time today to speak with all of us. Recently in a class I was taking, it was a genetics course, our professor presented us with an image, and it was a figure from a recent publication that I think actually was the All of Us data set that you were previously talking about. And they showed a U-map where it was connecting people's genetic information to that of their ethnicity. And there was a lot of backlash to this figure from both the public and from other geneticists in the field, questioning why they presented the data as this, because it's a really hot topic on, do your genetics represent your ethnicity? And there's a lot of confusion on that, and just a lot going on with the public with that. And I was wondering, for your guys' own work, how do you ensure that the way that you're presenting your data, especially like connecting genetics to race and socioeconomic, is not only misread by the public, but also not misconstrued by other people that have separate agendas from what you're trying to present?
Good question.
Yeah, absolutely. Well, yeah, I can start and then we can kind of go down the line. I think it's really important to know that race is very different from your genetic ancestry. Race is a social construct, and it's self-reported, and it can vary, based on how people self-identify. And that might not be the same as your genetic ancestry, which is really, just how your genome looks from, thousands of years of, people mating and going across societies and everything to form these kind of specific patterns that you see. And so from our genome, scientists can say, oh, it looks like your genetic ancestry dates back to, Western Europe or something like that, right? And so that's very different than saying, I self-identify as a white person. So I think keeping those separate is important, and can help kind of avoid some of that confusion.
Our group here, great question again. Our group here had a meeting just last week about this general topic because there was a National Academy of Science report trying to make sense of the question you're raising. And in our reading there was several recommendations about what not to do without recommendations of what to do. So an example of what not to do would be to do what Lauren said you should not do, which is to conflate self-identified race and ethnicity with these genetic measures. So that was thou shalt not do this. But there was just never a part in the report that we found to say what you should do. So I would say it's very unresolved now from our research understanding. And one convention has been, until very recently, that essentially every study that you would read about in the news, if there were people who did not have European ancestry in the data, so some data sets don't even have such individuals, but those who do, they were not analyzed. They were just kind of set aside because of this issue of we don't know what to do. What we do know that we can say that we're going to analyze this subset of the data who appear to have ancestry consistent with having relatives from Europe, and therefore some large share, it's not 100%, but it's close, of every finding that we know are from these subsets of the world, as both Lauren and Anya were saying. So we're really in a pickle there in some, even in some of the better cases of where we would like to bring these genetic predictors out in the wild for on the clinical side or even on the policy side, we have to know and own up to the fact that they're not predictive outside we can't assume that they're predictive outside of the population that the data is coming from. And in a society like the U.S., that's just very consequential. If we were trying to even get a genetic predictor of learning disability that where all the data are coming from white individuals and then bringing them to Madison Public Schools, which does not have only white individuals from them, we should be very understanding that that's very we should pause because there's not even what we should do here because we don't know what we should do. We should understand that some of these major problems in doing that, and so there's not just a public issue about whether everyone's in agreement or not. We should be doing that. There's a scientific problem that we cannot do that right now.
I mean, I will say it gets especially complicated when you try to translate it into the clinic. So I was a consultant on a project, a major NIH-funded project, where they're doing polygenic scores for medical, so cancer, heart disease, asthma, alike. And it's a translational funding project, and so they're actually returning results into the clinic. And they're returning to people of all sorts of different ancestries. And the predictive value of the scores are different for different populations. And they know that, and they have the odds ratio that says, this is this predictive for somebody of European ancestry, this predictive for somebody of African ancestry. And so the study, who's actually returning results, had to decide, what do we tell people? Do we give them the odds ratio or the predictive value that matches their ancestry that we know because we have their genetic data? Or do we then tell them their ancestry? What if the ancestry doesn't match with their identified race and ethnicity? What if they don't want to know their ancestry? So all of a sudden, they're walking into their doctor's office to get information about their risk of diabetes, and they learn that their ancestry is something they didn't even think about, right? And then they're like, that's not, right? So what the study ended up doing is providing the report saying, you're at, you have this score, you're in this percentile, and here's what that percentile means if you're European ancestry, if you're Asian ancestry, if you're African ancestry. Now, what does that do? It leaves out people whose genetic or whose self-identity don't match their ancestry because they might self-identify into the wrong that's the wrong way to put it, right? But they might not self-identify into the ancestry that their genetics match. And then what do you do for people who identify with multiple ancestries just based on where their families are from? And so when you roll this out into the clinic, it gets really tricky. And so that was a long discussion. And there was a paper just recently published about that process, that of the ethical decision making that went into that decision of just how to present that data to people. If you're interested, I could try to track it down.
Good question. Sounds like we have time for one more question. It looks like there's a hand in the back corner.
Now it's on. So building on this last point that you were just discussing, it seems to me that one of the things that handicaps this work is that we cannot extract ourselves from the social and political power systems in which we abide. And when we bring forward this information, we have little, I'm going to call it this way, we have little efforts to be able to control how others may use the information. And even, for example, in the case of Gina, if I felt that my data was misused and I wanted to challenge the organization with which I had provided that information, that would be me and my lawyer sitting next to a company and all of anybody that they want to put on that table. And so let's look at the power dynamic in that. And so I think we exist in a culture where the word is almost fearful of being able to manage those different capacities that folks have and turn around and protect that. And as you know, it's interesting, I've been thinking about this for some time and knew that I was coming, but I also have a neurological disease and I have children. And so, that disease has different kinds of genetic dimensions to it, potentially. And so it could be anything from a 75% chance my child will get it to a 10% chance. And so if I had that test out and they wanted to buy long-term care insurance, would you sell it to them? I don't think so. Not if you were this person with the insurance to sell. So I guess I'm just trying to say that we live in a context that I don't hear us talking about enough, and that is that social and political power and how that can really impact the options we may have and who has ability to stand up and defend themselves against misuse. So.
Yeah, I think that's well said, and I think it goes back to the balance, right? How do we prevent some of those misuses while helping to build up the positives of this research to move society forward.
I agree with that too. I guess one thing I want to say about context for us to keep in mind, because some of what I think some of our comments come across as more sometimes negative about this enterprise, but I think we should always, at some point in thinking about whether to do this, and this is going back to the first question about like where should we, what's promising here, is to think about what the current situation is in how we give one person treatment versus another, how we assign kids to kindergarten, how we our insurance practices and so on, because we're not starting from we're not starting from like everything's working perfectly now and then saying, well, these are all going to make everything worse. I think there's instances where, imperfect as it is, these new innovations should very well be on the table, clinical and otherwise. We're both, I think all our research is leading us to be cautious, especially on how some of these practices can increase disparities in the population, and always have that on the table. But there's instances, and you were talking about your own family, where this information, you could imagine instances where you would prefer that information being in the clinic or being at the schools or somewhere in their lives. And right now we just don't know when that's going to be on the table and when not. And I think that's part of why having this conversation today is both alerting that there's so much information. There's this fire hose. We have not dealt with it all at once. We don't have policies that are expansive. So to have these conversations now about in what instances we really are enthusiastic and others where we really need to be cautious. Thanks.
Well, thank you. So we're sadly out of time. This has been a fascinating conversation. Thanks to all of our great panelists for doing this. I certainly learned a lot. I assume most of you did as well. Jason wanted to make a point that if audience members still have questions, he and I think other fellow panelists will be around afterwards to answer your questions. So thank you to Follette for doing this. Thanks to all of you for coming. And thanks to our great experts for today's discussion.
Thank you.